to thank Professor Hisham Al-Khayyat for this kind introduction. Bismillah ar-Rahman ar-Rahim. It is my pleasure to be with you today and to share in this important conference, and I wish to thank the organizing committee, especially Professor Dr. Ines Shaltut and Professor Dr. Muhammad Naguib. My talk today will be about the new medication for HCV, what we call the direct antiviral agents, which is very effective in treatment of HCV, and we are looking for their role in fighting COVID-19. To talk about COVID-19, we know that coronavirus disease is an infectious disease caused by severe acute respiratory syndrome, coronavirus 2. So the virus is SARS-CoV-2, and this virus is a beta coronavirus positive sense RNA virus, where they depend on RNA-dependent RNA polymerase is a crucial enzyme in its life cycle. This enzyme is very crucial for the life cycle of the COVID virus. And also we noticed that both HCV and SARS-CoV-2 are positive sense RNA viruses and sharing the need for this enzyme for replication. And here is the point that we started from. About COVID management, we know that there is no specific treatment for COVID-19 till now, and we don't have an antiviral treatment for COVID virus till now. What we are doing, that we are making repurposing drugs to treat COVID-19. This is a short-term solution until vaccines or new experimental treatment can be developed. What we are doing, that we are take the drugs previously developed to treat other disease, as we are talking here about the direct antiviral agents for treatment of HCV, and to evaluate whether they can work against COVID-19 or not. And if they do work, they can be deployed immediately because this drugs is present in the market, they are safe, they are good for treatment of other diseases. And if we talk about the direct antiviral agents, we treated in Egypt more than 3 million chronic HCV patients by these direct antiviral agents, and we know that they are a safe drug, and at the same time the cost is good. They are not costly drugs. The WHO put some regulation targeting product profile for COVID-19 therapy, and the WHO said that the preferred is to have an effective treatment reducing mortality, or at least effective at reducing progression of the disease endpoints, including duration of hospital stay. And it is preferred to be once per day dosing and oral. If it is not, twice per day dosing oral or parenteral. And as we know, the direct antiviral agents can be taken once per day dosing and oral. And also the shelf life of at least 36 months and room temperature shipping, and this is also present in the direct antiviral agents. If it is not like that, the WHO is looking for drugs with shelf life at least 6 months. So this is the regulation that WHO recommended for having an antiviral drugs for COVID virus. Well, how we can evaluate these drugs, how we can know that these drugs like the direct antiviral agents are effective against COVID. There are many trials with a range of different endpoints. Some of this trial look for the viral clearance, and this is for phase 2 trials. Some are looking for clinical recovery for mild and moderate infection. Some are looking for 7-point severity scale, and this is for severe infections. And some of this trial is mainly looking for survival for severe infection. If we give this drug to a COVID patients and compare it to the other group and looking only for the mortality. For prevention studies, typically to use endpoint symptomatic coronavirus infection. When we go in more details, if we are looking for clinical recovery, 4 conditions have to be met after 10 to 14 days of treatment. We must have no fever or fever less than 37 centigrade, respiratory rate normal, oxygen saturation above 94. There is no cough or mild cough, and also it is very important to report if this cough is mild, moderate, or severe. And clinical recovery has to be maintained at least for 72 hours. So if we have this end stage clinical recovery, this is some point that we can look for that. Also for severe infection, as I said, we can depend on 7-point severity scale. What we mean by that, the best patients is not hospitalized, or if it is hospitalized, it is not requiring supplemental oxygen or even required any medical care except the per protocol administration. If it is hospitalized and need any medications but don't need supplement of oxygen, this means that he will have a 5 scale. If he is hospitalized and requiring low flow supplement oxygen, he will have 4 scale. If he is hospitalized on non-invasive ventilation or high flow oxygen device, he will be scaled as 3. Hospitalized patient with invasive mechanical ventilator will be scaled 2. And of course, if there is mortality, the scale will be 1. So this is a method to evaluate the response of the drug on patients with severe COVID infections. We have a lot of drugs that are evaluated as a repurposed drugs. From the antiviral drugs, we have Repurposed, which was used for influenza, hydroxychloroquine, which was used for malaria, nitazoxanide, used for treatment of Giardia or cryptosporidia, and also drugs used for HIV or SARS. And we have also another combination for drugs used for HIV. And we have the most famous drug nowadays, remdesivir, which was used for Ebola and SARS. Other strategy is to use dexamethasone, corticosteroids, or interferon beta as immunomodulator. This is, of course, not an antiviral drugs, or you can use other immunomodulators like tocilizumab. So what this happen if we are going to more in details with the direct antiviral agents? As we said that we have a lot of direct antiviral agents which were used before for treatment of HCV, and we have groups of protease inhibitor, NS5 inhibitors, and polymerase inhibitors. And under this group, there is a lot of drugs looking for this. And we noticed that sofosbuvir and daclitazivir, they are an anti-HCV direct acting antivirals, but it inhibits the RNA-dependent RNA polymerase that we said it is also important for the replication of the COVID. Daclatazvir also inhibit the HCV NS5A, and the studies showed that sofosbuvir and daclatazvir may bind to COVID virus. The first published study was published from Cairo University Faculty of Science, and this study depends on what we call molecular dogging. What is this molecular dogging? It is a computerized program, and it is now used for drug design for small molecules, and we are looking if the drug will bind to the site of the virus or not. And this study was done by our colleague in Faculty of Science, Cairo University, Professor Abdou El-Fiqi, and this study was published early in April 2020. And it had a lot of importance, and it was also looked for as the first thing that maybe these antiviral drugs for HCV can have a role for treatment of COVID. There is also another studies done in Vetro evidence looking for that sofosbuvir and daclatazvir are active against SARS-CoV in Vetro. And here we can see this study looking for the effect of sof and dacla on prevention or preventing COVID replication in Vetro. And here is also this study looking for the inhibition of virus replication, and we can notice that daclatazvir is more potent than sofosbuvir in inhibition of viral replication in these standard doses. This is why we started very early to put a study on clinicaltrial.gov looking for efficacy and safety of anti-HCV drugs in treatment of COVID-19, and it was planned to start in Cairo University under my supervision and our group in endemic medicine department. And at the same time, there is also another studies looking for the efficacy of sofosbuvir in combination with daclatazvir or velpatasvir on COVID-19. Why we are interested in that? All of us know that direct antiviral agents are safe to be administered based on our experience on treatment of patients with HCV. It can be used in patients with chronic hepatitis, including patients with cirrhosis and even renal impairment. The first abstract which was published, it was published in one of the virtual conference, AIDS 2020. It was about a study done in Iran. And also this study has been published on the Journal of Antimicrobial Chemotherapy. And in this study, they included 66 patients, 33 received the standard of care, and 33 received the standard of care plus also the sof-dacla combination. And the results was good. There was a good response to these patients who received sof-dacla, and we can look here for the time of recovery or hospital discharge. We can see that there is improvement if the patient is receiving sof-dacla in front of the standard of care. And also there is improvement in the arm who received sof-dacla as regard the mortality. Here the mortality is 5.4 compared to the control, which was 20%, and this was statistically significant. But of course, it is a small study. Those who received sof-dacla is only 33 patients. This is why the WHO needs more and more data. And this is why they didn't include sof-dacla in a big trial like Solidarity trial. And they asked us as a global group looking for the SOF and DACLA to start a big clinical trials looking for the efficacy of SOF DACLA in the management of COVID. And at the time of the consultation, there was no enough preclinical and clinical evidence to consider SOF DACLA to be included in large clinical trial like Solidarity. This was the data by the WHO in June. And this is why we started to collect more data from different countries, from South Africa, from India, from Egypt, and we are looking to have more and more data. Antiviral combination trials should include a fractional design to allow understanding which drug is driving the response and whether there is a synergism or interference between the drugs, and virological endpoints like viral load in studies in mild or early disease could be valuable for proof of consent. And here is the importance of having more data from countries with great experience in the usage of SOF DACLA like Egypt. What we did here, that we are running two big studies. One of them to look for the efficacy and safety of anti-HCV drug in the treatment of SARS pneumonia. And here, this is the specific objective, and this is the population. And we are looking for the primary endpoint is the rate of virological cure and the time to clinical improvement. And also we are looking for the clinical outcome. For this study, we are involving more than 4 centers, and we are aiming to reach 200 patients receiving the SOF DACLA in comparison to another 200 patients who are having the standard of care management. And this study, we hope that we will have the results as soon as 3 weeks from now. Also, we have another important study which is done only in Egypt. And in this study, we are looking for SOF DACLA as a preventive, because at the time of the epidemic, we have some of our patients receiving SOF DACLA not for treatment of COVID, but for the treatment of chronic hepatitis C. And we are collecting the data about these 1,000 patients who received SOF DACLA during the epidemic from March 2020 to September 2020. And look of these patients who received SOF DACLA for HCV have catched the infection by COVID, and if what is the incidence and what are the progression of the disease. So we are looking for data retrieval for these patients who received SOF DACLA for HCV during the epidemic, and we are making questionnaire for them to have if they have diagnosed or confirmed COVID or probable COVID, and look for the severity of symptoms and compare this group to other group who received SOF DACLA 1 year ago, that they have SOF DACLA before for HCV, but during the epidemic, they were not receiving SOF DACLA, and to compare this to the other group. So I hope also this study can end after 6 weeks. So we will have 2 big studies, one of them looking for SOF DACLA as a management for COVID, and other one as a SOF DACLA as a prevention for having COVID. So in conclusion, with the reproposing management, it is potential to develop a treatment with a proven safety profile and the clinical supplies ready to distribute to millions of patients, because we know that we have here manufacturing direct antiviral agents, so it's available, it is safe. And also we have to look for some of the dose going to be optimal, or we'll develop more doses or increase the doses. Can we develop these drugs and gain regulatory approval independently from large pharmaceutical companies? And also, what will be the future will double or triple combination. That can we use the direct antiviral agents 1 or 2 or 3, and also can we use it with some immunomodulating drugs. So this is a lot of studies and a lot of research that we have to do until we reach solve a problem of this very annoying disease, COVID-19. At the end, I want to acknowledge Dr. Andrew Hill from Department of Translation Medicine, University of Liverpool, United Kingdom, and also Dr. Ahmed Kurdi, Department of Endemic Hepatogastroenterology and Infectious Disease, Cairo University, for helping me in this presentation with some of their slides. And thank you all, and I wish to invite all of you to attend the USHID meeting from 5 to 7 November the next month. And this is the website that you can go to it, and this is the website, the personal website for me, where you can see this presentation. And thank you very much.